My name is Tom Lowery, and I'll take you through what we call the T2Sepsis solution and the two different panels that we have now in studies as also in clinical care. So the T2Sepsis solution here, sorry, there's a little auto advance here, is basically a complement to sepsis care. So our tests are rapid, direct from whole blood, and these tests are meant to actually help with the current standard of care, blood culture, and also therapeutic decision making. The key thing about the T2Sepsis solution is that it's direct from blood. So there's no waiting for a positive blood culture with this test. These tests go direct from a 4 ml K2 vacutainer. Then they're processed on the T2Dx instrument. You can see that right here. The T2Dx instrument has 7 drawers, random access analyzer. You can go and load a sample on there at any time that you need to. And then the result gets delivered anywhere between 3 and 5 hours. So there's many publications now showing that the time to appropriate therapy is now around 6 hours when using our tests. It's very different than the standard of care where blood culture takes multiple hours, multiple days to achieve a positive result. And so certainly there's a big advantage of speed. The other advantage with T2 magnetic resonance is the sensitivity. I'm going to take you through a variety of studies and show with you sort of the clinical sensitivity that's now proven for direct from blood culture independent detection with T2MR. We're in the 90% range up to 100%, and cultures in the 50 to 73% range. The current standard of care, I think everyone here is probably very familiar with for sepsis, at least the diagnostic piece of it, is taking a blood culture, and then when the blood culture comes positive, doing a gram stain, and then doing species ID, and then doing phenotypic or genotypic testing. There's certainly a lot of innovation going on in the area of rapid post-culture testing, but the therapy choices that are made for the patients is really tied to this diagnostic paradigm. Where patients are put on empiric therapy, and then the therapy gets adjusted at different points during the progression of information on what pathogen and what resistance profile their pathogen may have. And so our mission at T2 is really to accelerate that time to making those decisions to help inform the clinicians and the pharmacy in terms of choosing the appropriate therapy much earlier to help reduce patient mortality rates, help reduce hospital costs, and help reduce unnecessary use of antimicrobials. So this is the T2Dx instrument. The T2Dx instrument has 7 drawers on it. Each drawer accepts a different cartridge. It's a very simple and easy to use process for taking the 4 ml EDTA vacutainer onto the T2Dx instrument. And this is the actual workflow that's executed on the T2Dx instrument. And so I'm going to take you through the steps of those. So shown here on the far left, I'll use the pointer here. It's 4 ml EDTA vacutainer gets loaded onto the instrument, and the instrument actually selects for 2 mLs of that blood, subjects that blood to a detergent lysis, and then that gets pelleted, and then the supernatant gets discarded. So this is an important step in our process. We actually select for intact pathogen cells. So if there's circulating DNA that may be present in the patient sample due to the just standard immune activity to maintain the commensals in the right area of the body, that DNA will not cross-react with our test. It's only intact pathogen cells. And then those intact pathogen cells are subjected to a lysis step. That lysis step basically is a bead beating process, very efficient to a variety of types of pathogens. That occurs in about 50 to 100 microliters of a very viscous solution. So it has all the white cell nuclei, all the cellular debris from the 2 mLs of blood that was originally processed. It's almost a solid state at this point. And then that gets amplified. So we have a proprietary amplification method that works in that semi-solid state to amplify out the lyse pathogen cells. And if they are present, then we get an amplicon of DNA. And that amplified DNA gets detected by superparamagnetic particles that hybridize and cluster. So there's specific probes that need to be able to hybridize to that amplified DNA. That clustering process then is detected by T2 magnetic resonance. It's the same basic physics as magnetic resonance, so it's completely agnostic to all that debris that is present in the sample. And that's the key enabler here for our test. So basically just take the blood, concentrate it down, process it, and do the detections. Each species or resistance gene result that we provide requires a separate set of particles, so the sample actually gets split out after the amplification reaction, mixed with different particles, and the results get reported. Our results that we report are all qualitative, and so we have a cutoff on our T2 signal. If they're above the cutoff, then it's positive. If they're below the cutoff, it's negative. So there's a lot of data, and I'm just going to do a sort of a summary of some of the data out there now. Since we've had one test on the market now for several years, we have another test that we've just submitted to the FDA, and I'm going to share some of that data here as well. So on this slide is actually sharing some time to result data from the pivotal study we just finished for about 1,700 patients suspected of sepsis run on our T2 bacteria panel. You can see the time to result for T2 is very short. It's in the time frame of 4 to 5 hours. Time to culture positivity, the average is shown here, is 2 days. Certainly some come out sooner, some come out later. Time to actually species ID is 3 days, and time to negative result is 5 days for the standard of care. And so the data show that you get about 40 times faster time to result when you use T2MR as compared to blood culture. Also, a recent publication came out a few weeks ago using our T2 Candida test showing that patients are actually on appropriate therapy 8 times faster when the T2 Candida test is used as opposed to the standard of care. The other area where there's a lot of proven data is our clinical sensitivity. Now for 11 different species, we have proven clinical sensitivity in over 300 confirmed cases of infected patients, all in the ballpark of 90%, some higher than 90%, closer to 100%. If you go and look at the literature, what's the clinical sensitivity of a single set of blood cultures? Depending on the species, it's in the range of 50 to 73%. That's why we draw so many blood cultures to be able to capture the presence of that pathogen. There's also now a lot of proven data. I'm going to take you through some of it, and then the following speaker will also take you through more of it, showing that T2MR can actually detect invasive infections that culture misses, so that there's indeed intact pathogen cells in the blood stream of patients with deep-seated infections, and that can be detected by T2MR, really providing an opportunity to catch these infected patients much earlier with a higher sensitivity test. Specificity, so there's over 3,000 patients across 15 different institutions and several different studies now showing the specificity. On average, specificity is around 98%. For some channels, it's up around 99.9%. So this specificity is really important to be able to make decisions confidently in terms of what kind of therapy the patient may be on or may be held from. So there are now several papers and reports using the T2 Candida panel to actually withhold antifungal therapy or actually take patients off of antifungal therapy based on a negative result within the right patient population that has been shown to be appropriate and effective. Certainly these high specificities provide very high negative predictive values. And the last thing I'll just mention in my overview of data is there's a study now from a group in Spain showing that actually the T2 Candida test is a prognostic indicator of deep-seated infection, mortality, or clearance of serious infection that has about a sensitivity of 2.5 times higher than blood culture. Just a snapshot on the T2 bacteria pivotal study. So the T2 bacteria panel has 6 different species on it. It's essentially the escape pathogen panel, except instead of Enterobacteriaceae, we have E. coli. And you can see here the sensitivities for the different species, the specificities for different species, all in the ballpark range of what I've described, 90% or higher for sensitivity, 95% or higher for specificity, averaging out to those numbers you see on the top of the slide. So this is something we were really excited about. There was a question in the field of would we see similar clinical performance of our T2 bacteria panel as we do for the T2 Candida panel. The answer is yes, in terms of its clinical sensitivity and specificity. But one of the things we really dug into is we asked the question, what about those samples that were T2 positive, blood culture negative? So you can see over here, there's about 170 samples, because specificity wasn't 100%. There was about 170 samples that were T2 positive, blood culture negative. So when we dove into those, the first question we asked was, with the PIs on the study, were there other culture results from those patients within a time window of about 2 weeks when the T2 and the paired culture draw was taken? And if there was another blood culture that was positive for the same species identified by T2 Candida or another type of culture that was drawn, then we wanted to understand what those were. And so shown here is showing that about just over a third of these T2 positive blood culture negatives actually had other culture results from that same patient. Either a blood culture result, so 60% of them were blood culture results, about 30% of them were actually other culture results, demonstrating that T2 bacteria is detecting actually some of these T2 positive blood culture negatives are true infections, and about a third of those are actually infections that blood culture doesn't even catch, and the deep-seated infections catch. So when you net all those up, you get numbers like this. So overall, 39 patients the blood culture detected, T2 had all but 4 of those. Second tube was run on those ones and showed that with 2 of those 4, they were positive. The second tube showing that they were probably at titer levels right around the LOD of the test. And then here, blood culture missed a total of 63 patients that T2 identified that were actually indeed real infections. So that's where the opportunity here is that by using a T2MR-based test, you can catch these deep-seated infections that you're missing with blood culture. You can catch infections that blood culture catches much faster, and you can have a much higher clinical sensitivity to catch and treat these patients. A few case studies from the trial. This was presented by a group of Robert Wood Johnson, Mel Weinstein, and Tom Kern. So they were involved in our T2 bacteria study. So first case here is an example of a T2 positive culture negative. The patient had a history of VRE, bacteremia, and they were actually suspected of sepsis at the time. Culture came up negative, T2 positive. For clinical signs and symptoms, they felt like the patient was indeed infected, and the Klebsiella infection that T2 identified would have been consistent. Another case here is an Enterococcus faecium case that was positive on 2 sets of blood culture as well as urine cultures. They were then put on therapy. And then they came back and consented the patient for the study. In the study, T2 was positive for Enterococcus faecium, and blood culture was negative because the patient was already on therapy. Once again, this was counted against us in terms of our specificity, but another example of catching actually true infections with T2 bacteria. Third case is more of a classic case where a patient was consented for the study. Blood cultures came up positive after a few days for Staph aureus. They were positive for Staph aureus on the T2 bacteria test within a few hours. Just my second to last slide, I'm just going to touch on a study that was recently reported at ID Week. So 152 confirmed candidemic patients by blood culture that were enrolled in this study. The overall sensitivity of T2 in this study was 89%, so in the ballpark, again, of that 90% number I mentioned to you for clinical sensitivity. In this study, so this was led by Neil Clancy at UPMC and then Kevin Gary in Houston. They broke apart the 2 populations of patients based on whether they were not on antifungals or they were on antifungals. And I won't go through all the numbers here in the interest of time, but essentially the performance of blood culture in T2 were very complementary and similar in the patients that were not on antifungals. But those that were on antifungals, T2 was catching 10 times more patients that were infected than blood culture was. So one of the challenges of managing these patients, if you've already got them on empiric therapy, is you don't really know if they're infected due to the interfering effects of antimicrobials with blood culture. T2 doesn't have that issue. So in summary, there's a lot of data now building for using a culture-independent test like T2 magnetic resonance in patients that are septic. There's a lot of clinical evidence that it can actually lead to significant reductions in the length of stay. I didn't go into the details of the study, but there's been 2 studies now that have shown that by using T2 Candida in the ICU, they've seen 7 days reduction in the length of stay in the ICU. There's been several studies on saving money through the reduction in the use of antifungals for using T2 Candida, anywhere between $200 to $500 per tested patient. That's after the cost of the test. And then what we're really excited, though, is the opportunity to catch these infected patients earlier to help provide information to caregivers to be able to get the patient on appropriate therapy much sooner to help with the outcome of the patient. So with that, I will turn the time over.